Okay, so let's start with topic one. So I try to go through all of the whole slides, and these are your learning topics, learning objectives. Make sure you guys know everything on this list here. There will definitely be at least one question from each one of these bullet points, for sure. The one thing I didn't really go over is the 9 levels of organization. There's just a simple figure you guys can look at. It's very simple. I bet all of you guys already know that. You start with atoms, molecules, go to organelles, go to cells, things like that. So anyways, let's begin. Very simple stuff. You guys probably know this from biology. We have mechanism versus origin questions, or we have approximate question, which is a question that asks about the specific mechanism of a physiological process. And so simply all that means is how does this process work based on the laws of physics and based on chemical principles? For example, if we ask approximate question about those insects that produce light at night, we would simply just need to know that we have this protein luciferin and then ATP. You attach an AMP, use ATP to attach an AMP on it, and then you're able to produce light that way. So that would be an example of a mechanistic or approximate question. After that, we move on to questions that ask about the origin or also ultimate questions. That would simply be, why is this evolution advantageous? Why is this a necessary adaptation? On the exam, I'm pretty sure this would just be built into like an answer choice. So for example, you might get a question that says, which of the following statements answers an ultimate question, right? And so you'll get a list from A, B, C, D, E. All of maybe four of the choices will talk about molecular stuff, and then one of the choices will explain this is important because it allows them to breed and find each other at night. Things like that. So next, let's talk about multicellularity. Why is this an advantage? This is an advantage because most of your cells are shielded from the direct environment. And so the ones responsible for that, you should know this, the tissue, right? The tissue responsible for shielding your cells or your body is going to be epithelial cells. Epithelial cells are found between interfaces between different environments. So for example, the outside environment and the inside environment of your body. And then also it allows for specialization. Specialization allows for division of labor and an organism to become more complex. You can have limbs, you can move. For example, those light bugs, they can light up. And then there are disadvantages to being multicellular. Every time you have any process that becomes more complicated from other moving parts, if one thing fails, like for example, something in your car stops working, like the battery, the whole car will not work anymore. Very simple. And so the reason why multicellular cellularity evolved in the first place is because diffusion of nutrients and waste, they're only possible at small distances. And also, diffusion requires surface area to diffuse across. And so as over time, as organisms become more adept and they're able to get larger and simply just take up more room in their environment, you get problems where diffusion is not enough for the nutrients and wastes to be transferred in and out of the cell. And so that's what the problem is, right? Our biological mass, your volume, it increases at a faster rate than your surface area. And so specifically on in the exam, you could be asked to calculate this. Calculate the ratio between biological mass, aka volume of a cell, to the surface area, that's the surface area, to the volume. And I put these two equations here because these represent a sphere, and ideally, a cell is kind of in a spherical shape. But something important you should know is you don't have to memorize these equations. All you really need to know is that surface area is equal to radius squared, while volume is equal to radius cubed. And if you ever get any question on the exam, it might say, what happens when you double the radius of a cell? all you do is plug in a 2 in for r. 2 squared is 4. 2 cubed is 8. So what's the ratio? 4 to 8? That's 1/2, right? And you could also get another question asking what happens to the ratio if you decrease the radius. For example, let's say we cut the radius of a cell in half, plug in 1/2 for r, right? 1/2 squared, that's 1/4. 1/2 cubed, that's 1/8. And then the ratio to that would be 1/4 over 1/8. And then simplify that, of course, right? 1/4 divided by 1/8 is equal to 1/4 times 8, which is 2. I'm sorry for, I should have made a slide, but like I said, all you do is plug in whatever factor you're changing the radius by for r, and then you can solve the problem. And if you're still confused, at the end of this, you could ask in the breakout room. Next, conformity and regulation is also very important. Make sure you know this list here and you're able to assign which variables are most commonly regulated and which variables are more rarely, or not more rarely, but less animals regulate these variables, which ones belong to which group. And how does regulation work? Well, it's very simple. Regulation simply occurs because you have circuits in a cell, in the organ, or in throughout the whole body that function to stabilize this temperature in a changing environment. All right, and so if we zoom in on this graph here, right? So we have here a mammal that's regulating their temperature. If we zoom in on this graph and we change the x-axis to time, what ends up happening is we can see, small fluctuations like this. The body is never perfect. You'll never be at a perfect temperature. And the reason for these fluctuations, right, this harmonic wave, is because you always have deviations. For example, your body, whatever it's doing to produce the heat, for example, shivering or anything like that, it's going to produce too much heat, and then
I think I think your volume is, or we can't hear you.
sorry, hello, am I back?
Yeah, we can hear you
Okay.
now. All right, cool.
What did I don't know what I cut out on, sorry, but
Probably around like that deviation part,
okay.
negative feedback. Yeah.
So right here. So
Yeah.
as I was saying, every time you have a deviation, right, and you have some sort of variable that's being regulated, you're always going to be forced to return back to the set point. That's what this horizontal line means. And be sure that you understand this very specifically. This is what negative feedback is: a deviation that returns back to its set point. On the exam, you might get answer choices that will sound similar. That sounds like they're describing negative feedback, but make sure it's the answer choice would be something along this line. You have a deviation and you go back to the set point. All right, next we have positive feedback. And the definition for that is simply, rather than returning to your set point, you amplify the deviation away from it. So once again, make sure you understand the importance of this slide. It's simply just, these definitions are kind of strict in that sense. So next, some of you guys might have had problems with So for example, on an exam also, you might get a graph that looks like this. And like I said, you can tell this is negative feedback because there's a deviation, and then there's a return. And then also you could tell this is positive feedback because there's, instead of returning, you just go way off the graph. And on the exam, however, you also will get questions that ask you, is this positive or negative feedback? But you won't be given this graph, right? So instead, it's going to give you a block of text, probably something like this, or a circuit. I think in your, if you guys haven't checked it out, the figures preview for exam one, there was a circuit, but everything was redacted. So there's not much info on there, but make sure you guys know how to read circuits when I'll go through that now. So how do we identify if this whole system is a positive or a negative circuit or feedback loop without the graph? So it's very simple, and I broke it down into 4 steps here. Draw out the components, right? So literally, if you see ATP, write ATP. If you see a sensor, write down the sensor. Next, identify which variable that's being regulated. In this problem here, right? If you read it out, this was also in your, this example was in your practice, your extra credit homework. And then draw in your arrow, oh, sorry, the regulated variable is ATP here. And then draw in your arrows. Positive effect will be a regular arrow, and then if it has a negative effect, it's going to be a blunt arrow. And then finally, deviate your set point for, like I said, change this variable and then see if the circuit will make it go back to its set point or it's going to amplify it away. So let's begin, right? So if you read through this question, it gets kind of confusing if you read it too fast or you go over it too fast. The question here says muscle activity decreases ATP and the sensor detects low ATP. So at first you might want to write muscle activity, right? And then a negative effect obviously on ATP levels. But then when you get here, you get sort of a double negative that's kind of confusing, right? So it says here ATP decreases sensor activity. And personally, I would rearrange this to say low ATP instead. So that way we have muscle activity leading to low ATP, leading to sensor activity, leading to protein kinase A, B, and then whatever. So if you ever get a question on the exam, I would recommend you to change it. Just change the wording slightly. So instead of the double negatives, you can just do, you know, the arrows. So like I said, after that, we're going to deviate the set point to figure out if it's positive or negative regulation. We're going to increase muscle activity. Since it has a positive effect, we're going to increase this deviation here. Positive effect, increase, positive effect, increase, positive effect, increase, positive effect. And then here, right, sometimes the question might not specifically say how glucose transporters affect ATP level, but using common sense, you can understand that glucose is broken down into ATP. So what does that mean? Instead of reinforcing this low ATP level, we're going to be fixing it. We're going to be returning it back to normal levels, right? So instead of putting a up arrow here, we would put a down arrow. And so, as you can see, we deviated it on the first time around. And then what happens at the end of the circuit is we returned it back to our set point. So basically, that's all that you're looking for. If the 2 arrows are in opposite directions at the end, that means it's negative feedback. And of course, vice versa. If for some reason this arrow was the same as the deviation, if I decreased ATP, but then by the end of the circuit, it continued to decrease ATP as well, that would be positive feedback. And next, we have topic 2.
Okay, so I'm going to try and share my screen without it crashing. My computer's notorious for crashing when I share my screen. So I'm gonna start off with this is the outline of topics, but topic 2 is pretty lengthy, so I would say definitely go back through the PowerPoint. This is not a comprehensive and total review of topic 2. It just focuses on like some of the key points and some of the more difficult concepts of topic 2. So firstly, sorry, let me try and adjust this because I have like Zoom in the way of half of the slides. I don't know if you guys can see that. Somebody wants to like unmute themselves and let me know if there's like a
We
giant
only
square.
see the slide, so you're good.
Okay, perfect. Sorry, let me just shift this. Ooh, definitely not that. Sorry, guys. Okay, so the beginning of topic 2,
Excuse me.
just remember that. Sorry.
Will you guys be sharing the PowerPoint afterwards?
We didn't really talk about that. I don't see a reason why not, but
Yeah, I think
guess
we can. I'll send out a link with the recording, and then I'll also put a PDF of this presentation.
Thank you.
okay, so we start off with what can diffuse through a phospholipid bilayer is large, or like these cannot pass through, but what can pass through are small hydrophobic molecules like I think there's O2 hidden behind, yeah, O2, CO2, N2, benzene. So I would definitely memorize everything that does diffuse through. I'm not going to go through everything that does and does not diffuse through, but I would definitely know this graphic. And then these large polar and charged molecules, they use transport proteins to pass passively or actively through the plasma membrane. And then know that epithelial cells can either form sheets or tubes. And then sheets would be like skin or tubes would be like blood vessels. And know what like the apical and the basolateral membrane or side is. And the apical will face the lumen and the basolateral will face the opposite. Know that microvilli increase surface area compared to a smooth surface, and this increases absorption. Active transport uses ATP, so cells that do a lot of active transport will have a lot of mitochondria because they need the ATP. And then, like in another way put, if the cell has a lot of mitochondria, this indicates that it is a transport cell. Know that tight junctions are like stitches between cells, they seal them off. And I put a picture of that here. Desmosomes are like spot welds and they hold two spots of a cell together. And gap junctions form sort of like a tunnel between two cells where things can pass through. And some of those, I think I have it at the bottom here. I'm not sure if you guys can see this or if it's cut off for you, but it's cut off for me. Anything below 1000 Daltons can pass through these gap junctions, and some of these molecules can include ions, water, and some second messengers, but it has to be below 1000 Daltons. That is the limit for a gap junction. Okay. And then next, we're going to go over enzyme kinetics. So if substrate increases, then the reaction rate will increase until saturation or Vmax here. So that's basically what this whole graph is showing is substrate concentration. And as we increase substrate concentration, the reaction velocity increases until we reach saturation. Changing the enzyme concentration will change Vmax. So let's say I attach ubiquitin to some of my enzymes and they get degraded, then my Vmax will decrease. And that is that's the decrease. And just remember that enzyme concentration is related to Vmax. Substrate concentration does not change the curve, which is basically what I talked about before. If you increase substrate concentration, you're not going to change this curve, because that's basically what this curve shows, is substrate concentration versus reaction rate. So if we increase substrate concentration, that's not going to move this curve either way. And then changing enzyme affinity, Km, shifts the curve to the right or to the left. The substrate will either bind slower or faster, depending on if the affinity is increased or decreased. Vmax will not change if the affinity changes. And the way that I remember it is affinity, also known as Km, or like AKA Km, because I just associate affinity, like the first letter, and Km. And just to draw that out, let's say like I phosphorylate my enzyme and affinity increases, we're just gonna shift the curve this way, but Vmax will not change. If all we're changing is the affinity, it'll just reach Vmax faster, but Vmax will stay the same. And then a very important topic to remember is protein phosphorylation can affect Km or Vmax. It just depends on what the phosphorylation is doing. So it's really important to read the question and not just associate phosphorylation with one thing or the other. Okay, okay, so for acute processes that happen in milliseconds are phosphorylation, dephosphorylation, ubiquitination, and ion movements. And that's also up here. And then something that takes a little bit longer, which would make sense, is translocation of proteins and the secretion of hormones and other signaling molecules, because it takes time for it to like get released and embedded, or for secretion of hormones, especially if they're hydrophobic hormones, then we have to synthesize those, so that'll take longer. And then minutes to hours. To increase protein abundance, lipids, carbohydrates, and other biomolecules. Things that take days to months are changes in cell type, cell number, tissue size, and then generations are gene structure number and protein structure. Kinases catalyze phosphorylation. Phosphatases dephosphorylate proteins, and ubiquitin tags proteins for degradation in the proteasome. And transcription factors regulate transcription and protein levels. So transcription factors will go into the nucleus and they'll bind to whatever gene that they want to increase or maybe decrease. And then, sorry, I'm just gonna move that so that's not in the way. This is just like a quick little table synopsis of hydrophobic versus hydrophilic signaling molecules. Hydrophobic signaling molecules usually need to be translated and transcribed, so it is a slow process. An example is the reproductive cycle, and that's because hydrophobic, so if I want to store hydrophobic molecules in like a vesicle or something, these membranes are always going to be like phospholipids or something like that, and that's hydrophobic. So we can't store it because it would just diffuse right out since they're hydrophobic. So they're not stored, and we have to synthesize them every time we need them. So it's a slow process. The receptor location is usually inside the cell, and transcription factors might be an example. Common effectors are change in protein expression, because like I said, they usually hydrophobic will be, for example, in the nucleus. And so when transcription factors come in, they bind and they might change expression, or they might change transcription levels. And some examples are steroids, thyroid hormones, and gases. Gases are not really like synthesized or anything, but they are still hydrophobic. Then hydrophilic signaling molecules are usually stored in vesicles because they will not diffuse out. It's a pretty fast release. It's just exocytosis. The receptor for these are usually on plasma membranes. There's many possible common effectors, and some examples are biogenic amines and peptides. And then the 4 classes of receptors. First, there's a nuclear receptor, which is in the nucleus. Ligands are usually hydrophobic and diffuse through the plasma membrane and nuclear membranes, and they usually regulate gene expression. So this will usually be your transcription factors. Membrane receptors are on the cell surface. The ligand is usually hydrophilic and cannot diffuse through the membrane. And an example is ligand-gated ion channels and mechanically gated ion channels. G protein coupled receptors are usually on the plasma membrane, and a ligand binds to it, which will activate G protein, which then activates an enzyme or like a cascade. And some examples of GPCRs are smell, taste, and light receptors. And then there's enzyme linked receptors, which are also in the plasma membrane. The ligand or first messenger binds to the enzyme, which activates the catalytic site of the receptor and produces a second messenger. And an example of this is insulin receptors. And then one last important fact to note is that second messengers are important because they magnify or amplify a signal. One first messenger ligand can produce millions of products, and this helps save resources for the cell and fine-tune regulation. And then you want to know what a second messenger is. But one disadvantage of having a second messenger is if one thing goes wrong, then the whole cascade doesn't function properly. And then I'm not going to go through the details of this because it's pretty lengthy and it's pretty straightforward to just follow it. But you do definitely need to know this and all of these for the exam. And I believe that's the end of topic 2. Let me stop sharing. Oops.
Okay, I'll share my screen now. Tell me if you guys can see this, because I kind of have like not that great Wi-Fi right now. So
Yeah, we can see it.
It's all good.
Okay.
Yeah.
Okay, so topic 3 is about genomic approaches. So I'll be going over like the first part of topic 3 about forward approaches. So as you can see, there's 2 genomic approaches to studying gene function. There's the forward approach and the reverse approach. And I include this slide from his like PowerPoints because this is like a very important slide. It would be advised that you guys kind of know everything that's on the slide, like including like which are the forward approaches, which are the reverse approaches, and like what techniques go with which one. Because one of your questions will probably kind of mention one of these like in a subtle way, so you need to be able to identify which approach it is and which technique it is. And also you guys need to know how to distinguish between the forward and the reverse approach. The forward approach is basically going from the function to the gene. You need to be able to have the function first. So the function is usually like a physiological process that you know about already, and you want to know like what gene caused that process or that function to occur. And it doesn't have to be just a physiological process. It could be like a specific trait, phenotype, or a disease. It could also even be a set point. So basically just remember, you start with the function and you do things to find what gene is causing that function. So some examples of the forward approach. These are like examples of like how you could find the gene. So the first one is like with mutations and screening. I think in this approach, you would probably have like a trait that you want to study. And so you would create a bunch of different mutations in like a certain group of specimen. I believe in the slides he talked about mice. And you're going to see like which specimen's trait is changing. And it has to be like the trait that you're studying. So in his presentation, he talked about like the development of the head. So if you do like the different mutations and one of them is changing their head to have like 2 heads, then that's probably the mutation you'll be focusing on. And then you can sequence the mice's genome to find the gene that's been altered. And then the second example is with genetic diseases. This one is about like familial hyperkalemic hypertension. And this one is like kind of similar in that you can just sequence the individuals with this disease and those who don't, and then you can compare their alleles. And so the ones that have this disease would probably have alleles that are not going to be present in the people who don't have this disease. So with the forward approach, it's like a lot of comparisons. Okay. Okay, so then the third example is transcriptomics. This one, when you see this word, you should be thinking of like transcription. So that's why it means like focusing on analyzing the expression of mRNAs in like a certain group or population. So you should definitely know that this field of study uses like the techniques of DNA microarray and RNA sequencing. And they use these techniques to find like the genes that are being expressed during when a certain function is like going on. And then the fourth approach would be proteomics. This one is similar to transcriptomics, where you except it deals with proteins. So you're identifying protein expression or post-translational modifications that happen in proteins. Examples of like post-translational modifications that I think Isabella went over earlier, like ubiquitination or like phosphorylation. This field usually uses like mass spectrometry to do the protein expression, to identify protein expression. Okay, so now I'll just like go a little bit more in depth with the DNA microarrays. So with DNA microarrays, this is kind of like a more traditional technique. And you usually use this technique if you have the genome of like the group, the specimen you're studying. So you basically have 2 groups. One's going to have like the condition, the disease, or like the trait that you're examining. And then the other one is like a control. They don't have like whatever condition you're studying. So you take the mRNAs from both group, and you use a microchip. A microchip has like a bunch of different spots, and you can put like short pieces of DNA into these spots, and the mRNAs are put into each spot right after. So these oligonucleotides can base pair to the mRNAs, and they're usually very specific there's like different oligonucleotides, but they'll only base pair to a specific mRNA sequence from the specimen. So once they've binded, you can wash away these mRNAs, and then you can compare the spots that have the DNA that have the mRNA from the disease group, and then the mRNA from the control group, and then you can compare them and identify the differences, because the disease group that has their mRNA is going to bind to a different oligonucleotide than the control group. So that's how you can like kind of identify the differences. And then the last thing I think I'll be talking about is RNA sequencing. So with RNA sequencing, it is like easier to do this instead of like DNA microarray. But with this one, you're going to be comparing mRNA counts by just sequencing the mRNA from two groups. So you're still going to have the same two groups, the one that's like conditional and the one that's control. And when you do the sequencing, it's going to tell you exactly what gene it came from. So you can count like the number of sequences each gene has from both groups and identify the differences. So I have this example here that I did get from the slides, but basically you can see that they have pretty much the same number of sequences for every gene, except gene C. You can see like the first group has like only 5 sequences that like were counted for gene C, and the second group has 850. So that is pretty clear that the gene that they're differing in is gene C. And also group 2, the one with 850, is the conditional group. So it kind of indicates gene C has something to do with causing that condition. Okay, and I think that's it for me.
All right, I will be covering the second half. I'll be talking about model organisms and reverse approaches. So everyone can hear me all right, right? Okay. So for reverse approaches, it's the complete opposite of forward approach. So essentially, we're going to be starting with a gene, a protein, or a protein modification. And what exactly we're doing is we're going to manipulate that starting material, and we're going to see how these changes actually affect a phenotype, an observable process like a trait. And based on the changes that we observe, we're essentially going to make an inference of the function or regulatory mechanism of that gene we're trying to study. So simply speaking, we're going to be manipulating a target gene, and we're going to essentially determine the function associated with it. All right, next slide. Now I'm going to be talking about Krog's principle and model organisms. So according to him, he states that for a large number of problems, there will be some animal of choice or a few such animals on which it can be most conveniently studied. So simply what he's trying to say is that if you are trying to look into like a specific function, a specific gene, and trying to go deeper into research on it, there will be some sort of organism that we could basically utilize to expand on that knowledge. I'm gonna basically summarize all these bullet points. I don't want to burden you with all this knowledge, but basically what I want to say is that depending on like the physiological process you want to study, you can use like more simple ones like the C. elegans or the Drosophila or fruit flies, because they're accessible and cheap. They reproduce very quickly, and they're a lot easier to manipulate in terms of your genes of interest. So in terms of like research scenario questions that you're going to see in the exam, typically he's going to incorporate a lot of C. elegans and maybe Drosophila. But in terms of like the more complex physiological processes that are like more human specific, you would typically use like mice, which are a lot more expensive, slower in terms of reproduction, and it's a lot more of a methodical process. And there's also more ethical concerns related to handling mice. Next slide. All right, so for this first reverse approach, I'm going to be talking about transgenes and immunohistochemistry. So this approach mainly involves observing genes and proteins. We're not really manipulating the target gene, but we're utilizing this process to observe gene and protein expression. So what transgenes basically means is that we're getting a specific gene or a genetic material from one organism, and we're essentially sticking it in another organism. And we utilize this immunohistochemistry aspect, which involves these fluorescent labeled antibodies that bind to a specific protein. And because they're fluorescently labeled, they will essentially act as these markers that will allow us to determine where proteins are expressed in that alternate organism. So both transgenes and immunohistochemistry are going to work in congruence to help us analyze gene and a protein expression over space and time. So what I mean by that is that this approach will show you where a gene is going to be expressed or how gene and protein expression changes as an organism is developing, as shown in the picture on the right side. So next slide, please. For this next one, I'm gonna be talking about RNA interference. So this reverse approach actually involves direct manipulation of the genes and proteins. So RNA interference is also known as gene knockdown or silencing. So that's an important term that you should really know, because knockdown implies that it can get back up. So what I mean by that is that RNA interference doesn't necessarily alter the genome. It does nothing to the genome. What it does is that it actually destroys the mRNA transcript, which is basically the encoded message for a protein, but it leaves the gene associated with the mRNA alone, right? And without that mRNA transcript, you are essentially disabling the functional output of a gene. And because of that, no protein is formed. And essentially by preventing these genes from being expressed, you can essentially analyze how a physiological process or function may be affected in the absence of a protein. Next slide. Now I'm going to be talking about gene knockouts. So keyword knockout. So that implies that it can't get back up. So in terms of gene knockouts, you're going to be targeting a specific gene within an organism's genome. And with that gene, you're going to remove it or inactivate it, right? So you're just going to remove a whole section of the gene out of that genome. So unlike RNA interference, where you remove the interference, the gene can become functional again. In the case of gene knockouts, expression of the gene is permanently prevented, right? And the type of species that you're going to see in terms of like exam questions related to gene knockouts would most likely be mice, but you can also see some Drosophila or C. elegans related to it. Next slide. And for the last reverse approach that you guys should really know, it's related to CRISPR-Cas9 editing. So you don't really have to know the exact mechanism related to it, but I'll just provide you guys like a little bit of a basis. So in terms of this mechanism, what scientists do is that they determine a gene that they want to target, they want to edit, and they design this guide RNA that binds complementary to that gene of interest, right? And what they do is they get this guide RNA and essentially merge it with this enzyme known as Cas9. And what that guide RNA will do is it'll direct this Cas9 to that target gene. And that Cas9 acts as an endonuclease. So what I mean by that is that it will act like a set of molecular scissors that will cut out that target DNA. So with this technology, it's really important because scientists can edit practically any gene in the genome. So similar to gene knockouts, you can take a whole section of a gene out, but in the case of CRISPR, it's a lot more fine-tuned and precise, right? You can change a sequence of an amino acid codon, you can remove a gene, or you can insert another one. And that will essentially can render genes dysfunctional, or you can essentially activate a gene and express more of a specific enzyme. So this is a really important tool that allows us to determine when specific genes are turned off, like what happens in terms of the function. when specific genes are turned off in that organism. And I think that'll be the end of the content slides. So after this, I think we can break out into breakout rooms. If you have like any specific questions related to